So I'm going to cover basically 5 things very briefly in this short talk. And since this work has been going on for quite a long time, I'm trying to summarize a field that is actually pretty mature. So I'm going to just be brief and mention some review articles for some of these data. But in order to talk about pre-exposure and post-exposure prophylaxis, I feel it's important to mention the virus because there's an interaction between the two. Then how we've used these things to define effective cocktails and doses. What's the window of opportunity? What about ADA? I'm not going to talk about improvements in delivery. And then there's a little reality check at the end. So I messed myself up. I like to use this cartoon because it helps to illustrate the way we think about the virus and the ever-evolving envelope working together to drive antibody responses as the virus escapes from the selecting antibody. And it's a very small proportion of people who develop these very broad bNAbs. They do not control, even in these subjects, but these types of antibodies have been enormously useful to us. Prior to their, really the recent selection of antibodies like this, we were stuck with doing experiments with the polyclonal gamish. And the polyclonal gamish consists of non-neutralizing antibodies shown here on the left and neutralizing antibodies. And this is an incredibly busy slide. I refer you to this nice review by Amar Pegu and us, where we talked about some of the roles that these different types of antibodies may have. Suffice it to say that in the polyclonal gamish, we don't know which, if any, of these functions are taking place. When it comes to neutralization, we are fairly confident that at least one of the activities is to block infection and mop up things on the inside. And it's really those antibodies that I want to focus on today. You've heard about the structure of the envelope. This is just a quick reminder that there are several different places in the trimer that are vulnerable to attack by these bNAbs, and we now have a nice little arsenal of them. So that's what we've done with them. So this slide pretty much summarizes what we know about pre-exposure with passive antibodies. And the nice thing is that basically, if you give passive antibodies prior to exposure to the virus being any SHIV in your arsenal or potentially HIV, if we had such a model in mice, for example, you can block infection, and it doesn't matter which virus you're challenged with. In contrast, in the old days when we had access to the gamish, which had neutralizing activity, we could see some activity. We had an idea that neutralization was really important, but we really had a tough time sorting it out. So I think what we did learn from polyclonal studies was that neutralization is quite important and that antibodies can have an effect on infection. But it's been really a wonderful situation, and there are about 25 studies summarized in these 2 recent reviews that I think will help you if you'd like to read about all of those. What I think is important to understand, and we've talked about this today when we've talked about the differences between SHIV strains. Again, this is a very nice data from Amar's group showing that, John Mascolo's group showing that there's a big difference between the ability of specific antibodies to neutralize in vitro and in vivo, to block in vivo, depending on the virus. And when you take a tier 2 virus like SHIV SF163 and compare it to SHIV BAL, which is a tier 1 virus, there's about a 20-fold difference in vitro and at least a 10-fold difference in vivo. Why is that? Well, it probably is related to the rapidity with which the virus is getting into the establishing viremia in tissues. And to answer this question, we've done some studies, and I just want to, again, show you a little bit of data where we've looked at viral DNA in tissues, and we've also looked at viral RNA, obviously, in the plasma. Before I go there, I want to remind you of the history of what we've known about SIV for some time, which is both by oral inoculation in babies. A very nice study by Christian Salhenix back in 1999, and repeated also by Ruth Ruprecht and by Don Fedora's group, shows that there's very quick feeding of infectious virus, and you can't read this, but it's lymphoid tissues and then some gut tissues. And then there's a lovely paper from Chris Miller that Christina was also a major co-author on, looking at transmission intravaginally And again, you can definitely see that very early in infection, this is 2 hours, 1 day, 2 to 5 days, 6 to 10 days, et cetera. Whether you're looking in the genital tract, intestinal tract, or peripheral tissue, these are viral RNA. So we're getting a lot of early expression with SIV. And one thing we were interested in determining was with SHIV 162P3, which is a tier 2 virus, but typically SHIV, as you've seen, tends to control. When we infect infants by the oral route, we found very, very early signs of infection, day 1, day 7, day 14, with oral inoculation. And we could already see the virus in the blood, in the plasma, in the PBMCs. And importantly, as I'll show you in a few moments, this virus maintains an extremely high level of plasma viremia for the duration of the experiment. And we intentionally chose this model because we wanted this model, which we consider to be a really high bar with which to test the bNAb. This is to show you the virus goes directly to lymphoid tissues and gut. It's not in the B cell follicles in this animal. This animal has no T cell responses. It's basically a rapid progressor. It's being expressed in T cells, RNA, and DNA. Thanks to Jake Estes for this particular slide. But if you wait a little bit longer with SHIV P3, say 12 weeks of age, it starts to look like every other SHIV. You get these are examples of 3 different animals. There's a high one that's not controlling, intermediate, and a pretty rapidly controlling animal within really a couple of months. And this is proportional to the amount of virus that's in the tissues. The closed symbols are lymphoid tissues, the open symbols other tissues that are less concentrated. And we find that it's animals like this one that make strong, robust T cell responses and not animals like this. I won't say anything else about the immune response, but I want to just give you a quick glimpse again into this is SHIV 162P3 that I mentioned, the tier 2 virus versus the tier 1 virus. I think even without statistics, which we have, you can see that the peak viremia and the post-acute viremia at say week 9 is a log different at least, and the amount of virus we can find in tissues is clearly quite different using the same assay. So this virus is infecting for sure. It's infecting multiple tissues, but I would say that if we were to measure infectious virus, we'd have a tough time getting infectious virus from anything except lymphoid tissues in this infection, and many of these tissues we know from experiments we've done are actually positive. So Dan Baruch and Dennis Burton did a very nice experiment with Jeff and some other folks here. You may recognize your name here. I see Mark Lewis' name. This very nice experiment, which was actually a pre-exposure with PGT121 and adult female rhesus macaques, challenged with this same SHIV, and it was a short-term experiment that showed that you could block infection with this low but high enough to protect amount of antibody. But what was important about the study was they showed that the viral RNA was being expressed in tissues distal to the challenge site after the exposure, suggesting that the antibody has the power to get to the virus that's being expressed and participate in clearance. However, it is not known whether the antibodies actually did that. They have capacity to do that in vitro, but it could be that the virus just was neutralized and effectively reduced the dose. We've been trying to address these very same questions in newborns, and we're trying to do this in the post-exposure prophylaxis mode. So that for the next few slides, I'm going to talk about post-exposure prophylaxis with bNAbs. And again, this cartoon that's from a recent review that we wrote shows what, ideally, what bNAbs would do is they would either blunt the infection of different viruses, or they might well clear the infection, as was seen when the antibody was there first. And alternatively, polyclonal efforts to try to do this have been less than satisfying, to say the least. But again, indicative that antibodies have some role in modulating the infection. So we were pretty confident we could have some effect. And we published these data, so I won't belabor this point. This is a study we published in 2016 where we did a brief post-exposure multiple dosing with antibodies provided to us from the VRC very kindly. by John Mescola and Laura Pegu and their colleagues. And we gave the antibodies as a cocktail at multiple times in order to be sure to blanket this time frame. And we did serial sacrifices, and also we had additional animals that we took out. And again, using that same cartoon that I showed you before, we see that the antibody treatment really quickly clears the virus that starts to show up in tissues, and then we can't find it anymore. We are able to show, this is just a quick heat map to look at where's the antibody. Remember, this is a multiple dosing of antibodies, so this is not a kinetic study. But it does show that even 1 day after giving the antibody, you can already see that antibody can be extracted from tissue, and that it has some activity in neutralizing activities in vitro. So when you see these antibodies going up, it's because there have been multiple dosings. This says nothing about kinetics of distribution in tissue. We give all of our antibodies subcutaneously, but we do at least show that they're getting to a lot of different tissues, the same ones that we're looking at where the virus is. And we showed that, in fact, the virus is essentially can't be found after this treatment. Reminder that this is what the viremia looks like in multiple controls, and it's very tight. We just always see that we can get this very, very high sustained viremia. So we think that the ability to blunt and stop and clear that is actually significant in this model. At this point, we tried CD8 depletion and did not see any viral rebound. There's nothing in tissues. That's what this heat map is for, and it gives you a sense for what the heat map looks like in these babies. So this is, again, SHIV T3. So that's just a whirlwind look at the published studies. We have another study that we've just, that's in revision right now that looks at, this is a comparison of controls versus bNAbs starting at 24 hours that I just showed you. bNAbs that we've started at 30 hours using a single high dose, and bNAbs at 48 hours in the multiple dosing that I described to you. The goal here was we thought a single dose might actually be as effective as multiple dosing, and we wanted to see if we could reduce the anti-DNA antibody activity. And in fact, with multiple dosing, we're actually quite good at generating ADA responses against these are human antibodies in rhesus. So this is kind of a worst-case scenario, we think. These antibodies are all IgG1s, but they're human antibodies, and we do get very good both against CGT121 and against CRC07, and this affects the pharmacokinetics of the antibody in vivo, which you can see this one animal that had low ADA also is the one that had fairly long half-life. You've probably already looked ahead and seen that the single dosing did quite a bit better in terms of PK. The drop-off is reasonable, and about half the animals are not making ADA responses. So we're greatly encouraged by this. And what we did was we looked at tissues, and we showed that, in fact, the ones that are negative in the blood are also negative in all the tissues that we looked at. And then the animals that are in the controls are obviously very high. And then we had some animals that were intermediate with the treatment at 48 hours. And the question is, what do these levels of virus mean? And the student who did this work was Maria Shapiro, shown here, did a QVOA assay. And what she showed with this assay was that animals that copy numbers of viral DNA, which of course includes defective and full-length active integrated copies, not all of these can make infectious virus, but typically it has to be above at least 1,000 copies per million cells in order to be able to detect virus with this assay. So looking back at these levels, you might suggest that this is the kind of target we'd like to get to with remission. If we could actually drive down viremia to that kind of level, it might actually be helpful in the clinic. Others have been doing beautiful work, especially Malcolm Martin, with whom we've collaborated off and on over the years. This is a very nice study that he did with Michelle Nussensweig's antibodies 101074 and 3BNC117. These two antibodies were used also in a cocktail, and the antibodies were introduced 3 days after inoculation, either IR or IV. I think you can see the control here in gray, and then the IR challenged animals clearly were able to tightly control their virus, whereas the IV challenged animals, quite a bit more virus to deal with, are still doing much better than controls, but not as well as the IR. So, IR is a lot of copies, but there's a significant mucosal barrier to get beyond. And then this was a lower IV challenge, and there were no controls. So it's a little hard to interpret. But in general, their point was that in the animals that ultimately controlled their infection, they also made very strong CD8 T cell responses. And this is really not in conflict with our data. I haven't shown you those data, but we find the same thing. Animals that are not negative, but that have a little bit of virus, make very nice T cell responses. I've shown you one example of that. And to summarize, the time dependence of post-exposure prophylaxis with SHIV in monkeys using human antibodies, it appears that this is pre-exposure, obviously, but post-exposure, it works really well at 24 hours. We found that 2 different doses worked equally well. Day 2 looks, we got partial control with about 50% being tight controllers, and then another percent being a little bit better controlled. At day 3, when the Nishimura study, there was also partial control and some rebound in some animals with controlling T cells. We also, I failed to point out, but we also looked at CD8 depletion in our intermediate animals and also saw that we had strong CD8 responses and that we were able to, not on the graph. My apologies. I took the slide out. We also were able to induce virus. So we are in complete agreement with what they saw with their experiment. And then Diane Bolton had published in 2015 a nice study intervening at day 10 with the same antibodies. So we were using the same virus, SHIV-P3, and she found that control was at least as effective as ART, but there was viral rebound. But they did see limited seeding of cell-associated reservoirs, as we're starting to see. So I think there's a strong effect post-exposure, but the window is perhaps a little shorter. What about established infection? I just have a couple more slides to show you that this is a study from Malcolm Martin's group in 2013 in which they're comparing a dose of individual antibodies, and what's shown is the virus in red and the antibody in the colors, and there's a brief reduction of viremia in the presence of the antibodies, but then as soon as the antibody goes away, then the virus is rebounding, as you might expect with monotherapy, and this led to dual therapy and was undoubtedly the rationale for testing these antibodies in the system was in order to set things up for the clinical data that I'll show you in the next slide. And there is some significant duration of about 10 days or so in some animals. So there's the ability to see some significant viral suppression, but again, it's important that the antibodies be able to stick around. This was, again, about 10 mg per kg. When the clinical study was published at the end of last year, Mendoza et al., this is from Michelle's lab, it showed that at 30 mg per kg of each antibody, or 60 mg per kg total, these antibodies were given to infected subjects who had undergone ART suppression, and then right before ATI were put on this treatment. You can see that the ATI has driven the viral load down to the detection limit. Here comes the antibody, 3 different infusions, and hopefully you can clearly see there's good suppression of viremia, and there's a differential level of control, except for a couple of people, these 2 down here. Everyone appears to be rebounding as soon as the antibody goes away. This is a really beautiful paper. It's got a lot of information about escape variants and how the antibodies are contributing to selection. I want to just throw in a caveat, though, in all of these studies, this is very early clinical work, and these people were very carefully selected to be sensitive to these antibodies. So where are we with respect to conclusions? We think that early PEP may reduce viral reservoirs, and I haven't shown you, but spare T and B cells. There's quite a lot of damage in the model we're using where it's extremely high viremia. We think that single high dosing with antibodies could be useful for potentially a drug holiday, and that's what's suggested in the Mendoza paper. But we think that the data suggests that antibodies are likely to be most effective when they're present before the virus is widely distributed. And I've shown you the data. We did an experiment that I haven't shown you with a V2 antibody that showed we could not block infection effectively, but we could limit viral seeding. That's an open question. It needs a lot more study to really understand what was going on with that study. And long-term effects after ATI are really not known. In terms of the future and a reality check, the VRC01 AMP study is ongoing right now, and we should get some results from that study. VRC01 is also being tested in babies in conjunction with antiretrovirals, so we'll have to see. And I'm sure there are additional studies that Michelle and Mrs. Weig is doing. There's a lot more to come, and I think you're going to hear more about that in a few minutes from Amar. So I guess my caveat would be the same as we've heard all day, which is that be very cautious about SHIV in adult animals, but I think there are ways that we can use SHIV models to answer very specific questions. And I'm encouraged about the role for bNAbs in controlling viremia. So I'd like to just note my collaborators, and in particular thank Maria Shapiro, who's done the lion's share of this work, and Ann Hessel, who's my colleague in this study, and our funders. Thank you.